All right. Yes, we are recording today's lecture. You know how they say it's not brain science? Well, today it is. All right, so we are in Module 8. Come on. What's going on? The brain and the spinal cord. So we're looking at the CNS, the central nervous system today, all right, which includes the brain and the spinal cord. All right, so there are going to be a couple of little changes I have you make on your book because the way I give the lecture is not exactly the same way that the person who wrote the book has it set up. So I'll try to tell you along the way what I'd like for you to do. All right, first of all, we're looking at some general information. So on that first blank that says overview, go ahead and write down association neurons. And if you remember what association neurons mean, those are neurons that are touching other neurons, right? What other kind of neurons do we have? They're not touching other neurons, but they're touching what? Where's the synapse? Not between neurons, but between a neuron and a muscle. Okay, those are called motor neurons. All right, for motor, for moving. All right, but association neurons are the ones that touch other neurons. A lot of them are packed inside the brain. All right, there are trillions and trillions of association neurons, all, it says here, like together, but it's linked together. There are more, listen to this, more neuron-to-neuron links in one single brain, human, than there are electronic links in all of the computer systems of the whole world. Wow, it's pretty amazing. So our brain is so far superior to this thing, my computer right here, although sometimes it feels like our computer is smarter than we are, doesn't it? Well, our capability, our potential is much greater than this. Remember, somebody had to put this together. Somebody had to design this and program it to do what it does, and that was a human. The brain can perform in 100 steps what it would take the most sophisticated computer 1 billion steps. So our brain is amazing. Okay, it's this beautiful design that you hear that we don't use. We only use 10% of our brain or something like that. I don't know that that's true. I think we use all of our brain, but I don't think we tap into the full potential of the use of our brain unless we help develop it. So I'm kind of a believer in that whole use it or lose it kind of a thing, you know? So I think that, for instance, for older people, when they're starting, because we're losing brain cells, we know that, right? We know that we're losing millions every day, right? Yes. So how can we keep the ones we still have nice and healthy? I do think thinking strengthens your brain power. So I think elderly people, maybe if you give them puzzle books and things, if it doesn't, embarrass them or whatever. We did it for my mom. We'd give her these puzzle books, and it just keeps those things firing, keeps all those neurons going. All right, so the brain itself, that's what we're going to write down in this next set of blanks right here about the actual brain. It weighs 3 pounds, approximately. It uses 20% of the body's blood supply. Wow, that's a lot. So this little 3-pound thing in our head has lots and lots of blood running through it, around it, in it. Interestingly, you're going to find that it doesn't actually touch the brain cells. If blood touches the brain cells, it kills them. Wow, crazy. So it has to be protected from them. It's composed of 100 billion neurons at least. If you have something called hypoglycemia, it's when you lack glucose. And if you lack glucose, what else will you lack? It's the Jesus answer for this class. ATP, right? You got to have glucose to get your ATP. And if the brain is using 20% of the blood supply, do you think it needs lots of ATP? Mm-hmm. Lots and lots. The blood supply cut off, there's a word called hypoxia. Hypoxia happens when there's no oxygen getting to the brain. Okay, so if you don't have enough glucose in the brain, you get hypoglycemic. If you don't have enough oxygen in the brain, it's called hypoxia. And how can that happen? Well, the blood supply gets cut off via suffocation. All right, so if you withhold oxygen from your brain. You're suffocating. A stroke can do that. Okay, a stroke can do that by blocking a pathway for blood to get it to a portion of your brain. Okay, so a stroke is when you have a blood clot in your brain or in your neck that will cause the blood not to get to your brain. So those words are important, hypoglycemia and hypoxia. Of course, other things can cause hypoxia, drowning, and other forms of suffocation. Any way you're cutting off the air supply will cause that. And so that's indeed what kills you from suffocation, is the lack of oxygen, which you knew that already, but now you know why, because oxygen is necessary to make ATP. Right. All right. Are we good? Got something, each one of those blanks? So we're going to look now at something called stroke. I don't know if you've ever known anybody who's had a stroke. A stroke results from a blood clot or a brain bleed. So what is a stroke? A stroke is a blood disorder in the brain which causes there to be a problem. But there's two different kinds. Okay, so either the blood is cut off, so we have hypoxia to a portion of the brain, or the brain is exposed due to a blood vessel.
We are late.
Okay.
There was traffic.
If a blood vessel breaks, for instance. All right, so the two types of stroke. The first one is called the ischemic stroke. Results from a blood clot, either in the neck or in the brain itself. Sometimes those are called TIA. If they're very small, we call them mini strokes. It stands for transient ischemic attack. And this happens frequently as people get older, and they'll have a little, like what they call a brain freeze or something, and possibly a physical ailment of some sort. And they'll wonder about it. They can get a brain scan and show their little tiny plaques on the brain, which can show that there was a transient. Transient means changing. So that means they had a clot, but the clot went away. So that if you're going to have a stroke, I guess that's your best kind. The other type is called the hemorrhagic stroke, and that's if a blood vessel bursts in your brain. An aneurysm, it's called. So an aneurysm is usually due to a congenital defect in the blood vessel. In fact, in one particular area, it's right in the back of the head here, near the occipital part of the brain. If that blood vessel bursts, it bathes the brain tissue in blood, and blood is toxic to brain tissue. And so it starts to kill those cells off. So in a hemorrhagic stroke, so ischemic, you've got a clot. Okay, is that bad? Yes. All right, and we're going to look at the different types of things that happen. And typically when we talk about a stroke, if you've ever known anyone who's had a stroke, they probably had that first type, the ischemic type. And we'll look at those signs in a second. The hemorrhagic is a little more dramatic. When that happens to somebody and that blood vessel bursts, one of three things can take place. One, they drop dead right there on the scene. Two, they fall down and the ambulance comes to get them. They survive the attack, but they've got some pretty severe brain damage. Or three, they drop down, they come and get them in the ambulance, and they survive the attack and they have very little brain damage. Okay, so a third of them, I have a friend who this happened to. In fact, he was in a movie. He was watching a creepy movie, and he started feeling kind of this headache in the back of his head, and he went to the men's room. So he had his children with him, his adult children, well, teenage children. They were in college. And he left them in there. He went to the restroom, and he knew something was really wrong. And he laid down on the floor. And the next person that came into the restroom, he said, "Would you please call 911?" And they did. And they said, "My children are in the theater. I need you to let them know." And he turned out to be, fortunately, in that group of the 30% where he survived with little damage to him. But he did have that congenital defect which caused that aneurysm to take place in his head. After this, they had all of his children checked for that to see if they had it, and none of them did. So that was good. But his mother had died from an aneurysm. And so he kind of knew when he felt this. He felt a rush. He kind of heard a rush in his head, and he kind of felt like that's what was happening. So fortunately, they caught that. All right, so here are the two types. So when we have an ischemic attack right here, there will be, we'll see it says thrombus right there. Thrombus is another word for clot. and the clot happens right here. And look how many blood vessels are being cut off right there. And see, they're very blue. That means there's no oxygen getting to that part of the brain. Okay? So this is the one we'll talk a little more about in just a second. The hemorrhagic is the one I just told you about. There's this rupture of the blood vessel. You know, like I said, that congenital one that happens in a kind of in a general spot. But there can be other reasons why you have blood loss in the brain. What if there was some sort of trauma to your head or something like this that could cause a brain bleed? And that might even fall into that group known as a hemorrhagic stroke. So if you know someone, the signs of stroke are these: numbness or drooping of the face, arm, leg, especially only on one side. Okay, we're going to see in a minute how the brain is divided into two parts, two hemispheres. And that one side, the right side controls the left side of the body, and the left side controls the right side of the body. And typically if I have a clot, it's going to be on one side, and when that happens, the opposite side of my body will have these effects. Okay? So the numbness, drooping face, arm, leg on one side. Confusion, trouble understanding, trouble speaking, mostly because your face is drooping down on one side, yeah? And vision or loss, not necessarily loss of vision, but vision changes, but just in one side, one eye. Trouble with balance and a severe sudden headache. Any of these can be attributed to a stroke, and there are differences between men and women. So men, all of these that I just told you. Women, we add on to those a general weakness. I feel weak all the time, you know. What does it say? Disorientation and confusion, fatigue, and then nausea and vomiting. Interesting that that is more common in women who are suffering from typically a ischemic attack.
Yeah, there's a thing you use to check for a stroke, but there's like a synonym.
Here it is, fast.
Yeah.
This is it, right. So look, it's on your book there. Okay, there were a couple things you were writing down in that little chart part right there, but under fast, you're going to write down face. If you ask them, in fact, my mother, who just passed away, she was acting strangely, and I told her to smile for me, you know, because she was very sick, and she was in bed, and she smiled both sides. I said, okay, because I thought maybe she had had a stroke, because her right arm was very, very weak. But I made her smile, big old smile. I said, okay, we're cool. So ask somebody to smile, and if the one side doesn't go up, then you might want to call 911. Ask them to raise both arms. If they can't raise one, they just can't get just one side, that's a sign right there. Speech, so we have F-A-S-T, speech. If you ask them to repeat a sentence and it comes out very difficultly for them, right, and they can't say it. And then, of course, time, go ahead and call 911. It's not going to hurt, well, except maybe financially, to call 911. But if somebody's having a stroke, you can actually do something to help these people. And namely, what you can do is get them an anticoagulant, something that'll stop the clotting from happening. And therefore that clotting will quit. And after that, of course, if somebody has had a genuine stroke, they might need to be on an anticoagulant medication for some period of time, depending on how bad it was and this kind of thing. All right, so there's our acronym to ACT FAST. Okay, now we're moving on to brain tissue. We're going to look at the brain anatomy right now. Let's start with the tissue itself. So our first blank right there talks about gray matter. Gray matter. You may have had somebody tell you, my gray matter is not responding very well today. The gray matter is the part of your brain that does the thinking. Okay, it's the smart part. It's closely packed, interconnected nuclei, we'll get to that in just a second, what that means, and cell bodies of the neurons. Okay, so remember where the cell bodies are. Those are that part we call the soma. You've drawn that before, the soma. In the central nervous system, we call the chunks of gray matter, or little clumps of gray matter, we call them nuclei. In the peripheral nervous system, we call those little clumps of gray matter ganglion. White matter is the axons reaching out from the soma, and they're sheaths made of, the myelin sheath made of the Schwann cells, right? And in the CNS made of the oligodendrocytes. In the CNS, we call this white matter tracts. And in the PNS, we call those nerves. Remember how I told you the nerves were like a big, shiny, white shoelace? Okay, they're white because they're white matter. Yes. All right. I want you to take a look here. Okay, I'm going to go back up. So see my cell right here? See my soma and my axon? I'm gonna clump them together now. Ready? So here's another one. Here's another one. Do you see how all my soma bodies connected together? Their tissue is different from the tissue that's over here. All right, that's why this is gray. Literally, it's gray in color. And this is white because those sheaths and because of those myelin sheaths on there. Okay, you get it? So I want you to think of the gray like the computer and the white like the cables carrying the information somewhere. Okay, so these are my cables. Y'all all have probably wireless printers now. But think about, like, my wireless printer stopped being wireless, so I have to literally plug it into my computer every time I print something for y'all. That wire coming from the computer to the printer is like white matter. It's sending a message to my printer, telling my printer to do something. Print. Okay, you get it? So that's how we're going to keep it. We're going to think about it. Did everybody get what we needed on this page? This went into gray matter, white matter, and we're going to look at decussation here in just a second. All right. So that's the tissue, the underlying tissue. The decussation means to cross over. It means to cross over. All right, so there's a sentence down here. Before you see where it says decussation, leave that blank for now. It says the blank is the main part of the crossover from left brain to right side of the body. You're going to write medulla. All right, the medulla. Remember when we hear medulla, we think middle. Medulla is middle, so the middle part of the brain. And what's going on, you can see right down here, there is crossing over from left to right. Right here you can see it in the brain on this small picture on the left. You can see the crossing over right there. I want to show you this picture, however. Check it out. So I'm looking at, this would be the left side of the brain, because I'm standing here, right? So that's my left side of my brain. Look at all of the white matter traveling down. Do you see it traveling down to the other side? Look how much, about 90%. Okay, so it mostly crosses over. But there's about 10% that doesn't, that stays on the same side, right here, okay? But for the most part, it's this way. So when you've heard that left-handed people are right-brained, that would be accurate, okay? Right-handed people are more, their left brain is more active. And so we're going to look at those differences. The brain, y'all are going to dissect a brain when you get back in January. By the way, that first week you get back, you have a quiz right off the bat, right? A quiz on Module 8.
Wonderful.
And you'll be dissecting a brain, and that's due to this weird 16 weeks in our first semester thing, which is why we're having a lecture. In previous years, we would have finished last week. That would have been our last week.
Do you need a study guide?
In January, yeah.
Okay.
Yeah.
Because I already written it, I was like,
Yeah, we'll keep it so you can study it for the quiz. All right? All right, so now what you're going to write in the decussation blank, right over that right brain crosses over to left side, left brain crosses over to right side. However you want to write that. Put it that way. Decussation means to cross over. All right, we're going to be looking at the brain anatomy now. So are you turning your page? Yeah, you're turning your page. What I want you to do on page 49 is you should have something that looks like this. Yeah, 3 big boxes. And each one of those boxes wanted you to like draw stuff. And I'm thinking, yikes. So here's what I want you to do. In your first top box there, put a line down the middle. You're going to write down the 4 main parts of the brain on the left. And on the right, you're going to try to draw what you see on the screen right here. All right? So there are 4 main parts of the brain. One is called the brain stem. 2 is the diencephalon. Di meaning 2, encephalon meaning having to do with the brain. 3 is the cerebellum, and 4 is the cerebrum. So notice how similar cerebellum and cerebrum look and sound, but they're quite different, okay? Quite different. So now what I want you to do is you're just going to draw your, whatever you want to call that shape. What would you call that shape? Semicircle?
we're drawing this on the right side of the circle.
On the right side of that box right there, you're going to draw this semicircle, and then I'll give you a few hints. I'm going to pause this so we... Okay, hopefully you have a nice little drawing there. And we're going to go through now each one of these step by step. Okay, so What you're going to do in your second box, you're going to draw it in two. And on the left side, you're going to write brainstem. Brainstem. So, and you're going to take a few notes on brainstem. On the left side, yeah. You can write brainstem and you're going to write a few things down about it. You don't need to draw it again. But if we took it out, you would see it looks something like that. The brainstem is responsible for rudimentary functions. You know what rudimentary means? Very basic. For instance, rudimentary math. Can you add, subtract, multiply, and divide? That's rudimentary. So our rudimentary functions are part of what's known as the reticular activation system. It somehow regulates our sleep and awake cycle in there. I will tell you that we know a lot about the brain, but there's a lot we don't know about the brain. Okay, so there's still lots we're learning. The midbrain, so it's got three parts in the brainstem, three parts. The midbrain, which is right here in the middle, has white matter mostly. Okay, so mostly the brainstem is made up of white matter. So what is it doing then?
Transferring.
Exactly. It's the cable sending them the information, yes? Either bringing it to the brain or taking it away from the brain. But it does have some clumps of gray matter. We call those nuclei because we're in the central nervous system. There are four different ones. Two we think are for vision, rudimentary vision, light versus dark. Two for rudimentary hearing. Okay, not specifics, but general hearing. Then we have the pons, which is right here. Pons means bridge. Pont in French. Sur le pont d'Avignon. Pont, it means bridge. White matter for communication. There are some gray nuclei embedded inside here. We're going to look at those here in just a minute. And we believe those are responsible for inspiration, taking a breath, exhalation, releasing that breath. Do you think that's a rudimentary function?
Yeah.
Yes, we need to be able to breathe. And then we have the medulla oblongata, my favorite part of the brainstem. Some people refer to it as the medulla. What's happening in the medulla? The decussation, right? So we talk about that medulla, the middle. It's in the middle, but it's also in the brain stem. All right, so the medulla oblongata, the vital functions of heart rate, respiration rate, blood pressure, swallowing, and vomiting. Do we need those things to stay alive?
Yeah.
We do. It's kind of interesting. People don't think of vomiting. That would be one of those rudimentary functions of our body. All right, we've got to get rid of something. Swallowing. If you can't swallow, you can't survive. All right, so I have my little brain models here. Two of them.
What kind of brain are we dissecting?
It's a sheep brain. Yeah. All right, that one's a puzzle. The puzzles pieces are not put together, but it does show us something really interesting. Okay, so this shows you all the parts inside the brain right there. I believe those are labeled, but I wanted to show you this is the brainstem right here plus the diencephalon right here. Okay, diencephalon, brainstem. Although if you open it, the brainstem goes up in here, the diencephalon comes out like that. Okay, you'll find that. You'll dig that out of your brain in January.
Do we each get our own brain?
We're going to share a brain. So you'll each get a side of a brain. You'll start with it whole. You both cut it in half, and then each of you will take a half. Yeah, brain surgery.
Wonderful.
Yeah, it's pretty good. It's pretty cool. Okay, so on the second side of that box, you're going to write diencephalon. The diencephalon. Between the brain. Encephalon, I thought di had something would do it too. I think two sides. The two sides.
When you first said like die, like the first part, I was like, so we died?
No, the encephalon happens to come after it. This does spell the word die. Okay, there are two parts. There are the two things we want to talk about. Okay, the first one is called the thalamus.
You'd be a terrible quarterback, though.
This part right here, y'all, is called the thalamus.
The what?
Thalamus, right here. It's a large relay station. Remember, this part is also mostly made up of white matter. White matter. No, no, you don't have to draw it. You're just going to write these things down on the right. The thalamus is a large relay station.
Let me grab your cup.
Okay.
It's a good thing you didn't fall, Amanda.
There are sensory axons for a crude interpretation of the sense of touch. Motor neurons control the lower body movement, and we have some sort of mood control, including fear and anger, something like that in the thalamus. There's quite a bit of it. It's still quite a mystery about the thalamus and the hypothalamus for that. The hypothalamus is the other part. Okay, so the thalamus and the hypothalamus. It's poorly understood. We believe there's some control over mood and the regulation. The pituitary gland hangs right off of it. So you see the green part right here. The little pituitary gland is that little part right there. Hangs off of it and controls the autonomic nervous system, which includes fight or flight. If you remember our chart we had that you'll need again for Module 9, we're going to talk about that part, the ANS. So there's some sort of control of the body temperature in here. There's been some experiments done on rhesus monkeys, and with these monkeys, what they have done, they put a probe into the hypothalamus, and they made the probe very cold. And these monkeys didn't have any change in the environment they were in, and they started to shiver. So just by making the hypothalamus cold caused them to get very cold. Meanwhile, when they heated up the probe, it made them very hot. They started to pant, even though the temperature in the room they were in did not change at all. So there's something to do with temperature control in the hypothalamus. Also right here, see I said the hunger. You know how when you're feeling really hungry? Okay, my kids still do this to me to this day. If, say, you're driving on a big trip somewhere and you're like starving, you're like, oh, Mom, I'm really hungry. Well, we're not there yet. Wait till we get there. And it takes so long to get to the McDonald's. By the time you get there, you don't feel as hungry anymore. You're like, eh. You know, people will say something like, if you have a craving, wait for 15 minutes and it'll pass because you're not as hungry anymore. That's because it's the hypothalamus that's up there saying, feed me, I need glucose.
Right here, right now.
Now, give it to me. And when you don't feed it, your body, then the hypothalamus says, they're not going to feed me. I'm going to go instruct the liver to release some of that stored glucose. And that's what happens. And so my kids would say to me, Mom, my hypothalamus is about to give up on me. I'm so hungry, my hypothalamus is going to give up on me. I'd be like, that's good. We're keeping and driving, you know. All right, so you have the most important thing on this slide is you know the two parts are the thalamus and the hypothalamus. All right, next comes the cerebellum. It's that little thing hanging off right here. When I say hanging off, it is quite separate. It's like a separate little tiny brain over there. And you'll notice it when you do your dissection, it's definitely a separate part. So it kind of almost falls off a little bit. So you can see on this little model, it's this part right here. In my plastic model, which we'll eventually have to put together, it is a different color, and it looks like this, and it hangs off the back of the brain. Has two sides, because the brain has two sides. So when y'all cut your brain in half, you'll at least get a half of a cerebellum as well. All right, so what is the cerebellum for? It's responsible for subconscious voluntary motor functions. What is a subconscious voluntary motor function? So it's voluntary.
Are we still writing this in the same way?
Right now you're writing with your hand, right? And you're not really thinking a lot about that. You know, if you're like Gabby's tapping her foot, she's not thinking about that, but your muscles are doing that, right? So it's kind of this subconscious voluntary motor function. It's wait a minute, where am I putting this? On the next box down, you're going to draw it in half, draw a line down the middle, and you're going to write cerebellum on the left side. You're going to write cerebrum on the right side. All right, it has something to do with muscle tone, equilibrium. So when I'm standing here, it's my cerebellum that helps me stay standing in a normal fashion. I'm not falling over. Of course, dizziness and stuff, we'll look at that later when we look at senses. Coordination, the sequencing of muscle contraction. Okay, so I don't know if you've ever seen anybody who when they run, they look kind of goofy when they're running. Okay, it might be that their cerebellum isn't as sequence muscle contraction as well. Also, we have this thing called muscle preset. The example that's given in the book is if you go to pick up a carton of milk. So let's think we're talking about a carton of milk that's not in the see-through bottle, okay? And you can't see through it. And you think it's full, so you go to pick it up, but then you realize it's not full at all, and you do this. So your brain is thinking, I know how much this weighs, and when you pick it up and it's not that, you go up. So that's called muscle preset. And we believe that's taking place in the cerebellum, the control for that. And also the dampening. So it talks here about pendular arms. So when we run, we tend to do our arms like this. Yeah, we're running nice. But if you ran and let your arms flail around like a rag doll, okay, then that's really goofy looking, right? So we think the cerebellum has something to do with keeping our arms from just flopping around. Okay, we're keeping our arms like we get. It's the TV show where the guy runs with his hands like this.
Oh yeah.
Moe's. Moe's.
Yeah.
Okay. Yeah, it's kind of funny that way. So I kind of think of the cerebellum as being that thing that shows how coordinated you are muscularly. Okay, so like you are coordinated to do things like, say, gymnastics or something like that. My coordination's not great. I remember we had this project where we were killing chickens for food. You know, and we had to chop their heads off. And I'm sitting there going, oh my goodness, because my coordination, I'm gonna miss. I'm gonna miss. You know, so I was very not confident about cutting off a chicken's head. I didn't want to do it anyway, but yeah, I made somebody else do it. But the cerebellum's responsible for that, so I blamed my cerebellum. In fact, if somebody has a cerebellum injury, because it's actually kind of tucked up under the brain a little bit, so if you hit the back of your head, you tend to affect your occipital part of your brain, that's your vision center. But if somehow way beneath that cerebellum gets damaged or otherwise, you know, just rattled around or something like that, then somebody can be walking around like they're drunk. You know, their coordination's just gone. And we believe that usually has something to do with a cerebellum injury or something like that. All right. Next comes the cerebrum. That's in the right box over there. The cerebrum is the diva, the star of the show. This is where the thinking takes place. Of all the animals in the animal kingdom, no animal has the size of cerebrum that we have as humans. Our cerebrum is way bigger. Now, we do have animals with large brains, but their brain doesn't have as much cerebral tissue associated with it. Notice how it's wrinkly. That's important. We call that a convoluted surface. And so if somebody tells you you have a smooth cerebrum,
That means you're dumb.
they're calling you dumb. Yeah. So, but if they say, oh, I'm very convoluted cerebrum, you're smart. So this is where your smarts come from. All right, so it's the largest, most recognizable part of the brain. The cortex is the outer layer of convoluted, look, gray matter. Convoluted gray matter. Gray means it's doing the thinking, right? The folds that go in are called gyri. Some people pronounce it gyri.
For gray?
No, it's just
Oh,
the
okay.
fold. It goes down like the little valley part. And the fold, the part coming up is called the gyri. A single one we call the gyrus, or gyrus. And then the grooves are called the sulci, the sulci. One would be a sulcus. So that's the groove going down. And it's divided into two lobes, left side and right side. And we're going to look at those separately here in a minute. We're going to also look at it's divided into hemispheres. It's also divided into lobes. Those are these parts of different color right here. All right. Interestingly, those lobes, we have designated those. When you get your brain, it's not going to have different colors on it to show you where those lobes are. So it's not arbitrary. We put those lobes are where they are based on function. But you can't really tell by just looking at the brain. I think that little model you're looking at there does divide by color, does it not?
Yes.
Yeah, different parts, they're the different lobes. Those different colors are the different lobes of the brain. Okay, so on your next page, that means that page is full, right?
Yeah.
On your next page, it says additional notes. Next to that word, additional notes, right next to it, write lobes of the cerebrum. Lobes of the cerebrum. And then you're gonna go down those lines right there with each one of the lobes. There are actually 1, 2, 3, 4, 5 lobes, and there are 5 blanks. 1, 2, 3, 4, 5. Yeah. All right. So the first one we're talking about is called the temporal lobe. It is responsible for. Let me make sure. The temporal lobe. I just want to tell you where it is. I don't have a picture
It's
here.
in like the middle part of it.
Yeah, the picture will come up, y'all. Leave a blank there for it. It is responsible, we believe, for abstract thought. What is abstract thought? What does it mean to be abstract? It's the opposite of
Is it
concrete.
Like sort of abstract things like feelings and
Yeah.
imagination.
Things that you don't really see necessarily. If you're going to be thinking about thinking, you're going to be thinking about something that's not physical that we can touch, right?
Air.
Yeah. Well, but air is physical, you know. So, but if you're thinking about philosophy, philosophy's a good one. Very abstract. So abstract it gives me a headache to talk about philosophy. Okay. Religion. I mean, if you're talking about, people say, "Who's your favorite philosopher?" I'm like with George W. Bush. Jesus Christ is my favorite philosopher. Best philosophy on the planet, Christianity. You know, people will laugh at me, but that's how I feel. It also has what we call the primary auditory area. So here is where we get the basic sound sensations, pitch, volume. It also has the auditory association area. Okay, what this means is memory and context. For instance, if I played you a sound of a trumpet playing a music, you would say, "Oh, that's a trumpet." Why? Because you know it, you've heard it before. This part of your brain, of your temporal lobe, remembered that that's what a trumpet sounds like. And it might even bring it back a memory of that time I was at the such and such place and I heard the trumpet playing, something like that. There'll be memory associated there. Okay, the Wernicke's area. We're going to be looking at the Wernicke's area in a little more detail. This is where there is a little confusion about it, and there's been some changes on Wernicke's recently. The idea, but the speech organization and comprehension takes place, we believe, here on the temporal lobe in Wernicke's. And then something called the hippocampus. This is somehow responsible for memory, especially names, memory of names, recognition. Yeah, if you aren't good at remembering names, maybe your hippocampus is. Oh, and also our taste area is here, our taste area. The taste also involves memory. I've tasted this before. I know what that taste is. Okay, so here we are. The temporal lobe is coming along the side. And keep in mind it's on each side. Each side. Sometimes I get it mixed up with this one up here, the blue one, which is the parietal lobe.
So this is the one in the front? Oh, wait.
No, it's the pink one right here.
Okay.
Okay, so it's on the sides, come down by your ears, sort of like that. Okay? Interesting look, auditory area. See, it comes down. Your ears would be about right there. So I just wanted you to know, if you write on the temporal lobe on the side. All right, let's talk about the hippocampus. So the hippocampus is in the temporal lobe, but it's kind of running alongside like this. That blue part right there is running along on the inside of that temporal lobe. It's part of what we call the limbic system. So that's going to help us with memory and things like this. It's considered transitional between the brain tissue right there. It's between gray and white. Okay. Functions would include, we think, memory consolidation, integration of emotional tone with higher cognitive functions. Cortex provides semantic influence. Okay. Hippocampal memories, behavioral inhibition, inhibitory influence of brainstem activity. Okay, so behavior is going to help you behave yourself. It's called the hippocampus because people believed it looked a lot like a seahorse. So hippocampus really is referring to a seahorse. All right, so I think I have a video on this one. Yeah.
On September 1st, 1953, William Scoville used a hand crank and a cheap drill saw to bore into a young man's skull, cutting away vital pieces of his brain and sucking them out through a metal tube.
Yikes.
But this wasn't a scene from a horror film or a gruesome police report. Dr. Scoville was one of the most renowned neurosurgeons of his time, and the young man was Henry Molaison, the famous patient known as HM, whose case provided amazing insights into how our brains work. As a boy, Henry had cracked his skull in an accident and soon began having seizures, blacking out, and losing control of bodily functions. After enduring years of frequent episodes and even dropping out of high school, the desperate young man had turned to Dr. Scoville, a daredevil known for risky surgeries. Partial lobotomies had been used for decades to treat mental patients, based on the notion that mental functions were strictly localized to corresponding brain areas. Having successfully used them to reduce seizures in psychotics, Scoville decided to remove HM's hippocampus, a part of the limbic system that was associated with emotion, but whose function was unknown. At first glance, the operation had succeeded. HM's seizures virtually disappeared, with no change in personality, and his IQ even improved. But there was one problem. His memory was shot. Besides losing most of his memories from the previous decade, HM was unable to form new ones, forgetting what day it was, repeating comments, and even eating multiple meals in a row. When Scoville informed another expert, Wilder Penfield, of the results, he sent a PhD student named Brenda Milner to study HM at his parents' home, where he now spent his days doing odd chores and watching classic movies for the first time, over and over. What she discovered through a series of tests and interviews didn't just contribute greatly to the study of memory, it redefined what memory even meant. One of Milner's findings shed light on the obvious fact that although HM couldn't form new memories, he still retained information long enough from moment to moment to finish a sentence or find the bathroom. When Milner gave him a random number, he managed to remember it for 15 minutes by repeating it to himself constantly. But only 5 minutes later, he forgot the test had even taken place. Neuroscientists had thought of memory as monolithic, all of it essentially the same, and stored throughout the brain. Milner's results were not only the first clue for the now familiar distinction between short-term and long-term memory, but showed that each uses different brain regions. We now know that memory formation involves several steps. After immediate sensory data is temporarily transcribed by neurons in the cortex, it travels to the hippocampus, where special proteins work to strengthen the cortical synaptic connections. If the experience was strong enough, or we recall it periodically in the first few days, the hippocampus then transfers the memory back to the cortex for permanent storage. HM's mind could form the initial impressions, but without a hippocampus to perform this memory consolidation, they eroded like messages scrawled in sand. But this was not the only memory distinction Milner found. In a now famous experiment, she asked HM to trace a third star in the narrow space between the outlines of two concentric ones, while he could only see his paper and pencil through a mirror. Like anyone else performing such an awkward task for the first time, he did horribly. But surprisingly, he improved over repeated trials, even though he had no memory of previous attempts. His unconscious motor centers remembered what the conscious mind had forgotten. What Milner had discovered was that the declarative memory of names, dates, and facts is different from the procedural memory of riding a bicycle or signing your name. And we now know that procedural memory relies more on the basal ganglia and cerebellum, structures that were intact in HM's brain. This distinction between knowing that and knowing how has underpinned all memory research since. HM died at the age of 82 after a mostly peaceful life in a nursing home. Over the years, he had been examined by more than 100 neuroscientists, making his the most studied mind in history. Upon his death, his brain was preserved and scanned before being cut into over 2,000 individual slices and photographed to form a digital map down to the level of individual neurons, all in a live broadcast watched by 400,000 people. Though HM spent most of his life forgetting things, he and his contributions to our understanding of memory will be remembered for generations to come.
Okay, so we're still doing research on that kind of thing. Next, we're going to be looking at the occipital lobe. So the occipital lobe is your next blank. It's at the back of the brain right here, this green one on this picture right here. It's at the back. It's the visual cortex, which gives us the basics of vision, shape, color, size of whatever we're looking at. But it also has the visual association area, memory, context, history, recognition. Okay, the idea that I see someone, I know it's you. Okay, so that's in a different part of the occipital lobe. All right, context, I know where I saw this before. History, recognition, all that memory associated. But over here, basics right here, shape, color, size. All right, and of course we know that we're going to talk about the occipital lobe more when we learn about vision. And that's going to be in Module 9 when we're talking about the peripheral nervous system again, looking at all of our senses right there. Bless you. All right, I'm going to move on, y'all. Otherwise, we're not going to finish this. So if you need me to send you the lecture, I'm recording it. And you can do that, and you can pause it to watch stuff. Okay. This is a condition called face blindness. It's pronounced prosopagnosia. Prosopagnosia. And this is a condition, so oftentimes it happens when somebody's had some damage to their brain and they see a face and they can't recognize it. They don't know who it is. And you can imagine how awful that would be. That every day you wake up in the morning and you see your mom, you don't know it's her because you don't recognize her face. It's blurry, but it's not really blurry so much as you see a face and you don't know who it is. And oftentimes those people have to learn to listen to a voice so they know who they're looking at. Okay, so brain damage can cause that. Parietal lobe. Okay, the parietal lobe is the one that goes over the top, right? So over the top. It's responsible a lot for our senses. We have the primary somatic sensory area. And if we're looking at the brain, I have my little pink model here right now. Do you see how I've colored in this gyrus? Okay, the sulci and the gyrus, right? So this is found, so the primary somatic sensory area, all of our senses are located on a single gyrus. This one coming out here, it's called the post-central gyrus. So it's right here, here's our little line, post-central gyrus, and you can see all along this little part of the brain right here, every bit of our sensory reception takes place right there. Pretty amazing. It reaches over the whole body naturally. It will tell us a localization of sensation. So for instance, if you have a bee sting, you know where it is, right? It's on my leg, it's on my arm, whatever it is. So it localized tells you where it is. So we have had people who have donated or have volunteered to go into surgery. They're awake when they go in, and they will go in and touch parts of that gyrus, and they'll say, oh, I feel that on my toe, I feel that on my... So we've mapped the whole thing. Isn't that crazy? And not just feeling, all senses start there. Let's see. Somatic sensory association determines the nature of the sensation and memory of it. So if it's a massage, you're like, I like that, it feels good. If somebody punches you, I don't like that, that doesn't feel good. Okay, so the memory there. That's all in that parietal lobe that kind of stretches across both sides right there. All right, there it is in the green section going across the top. All right, the frontal lobe. What's your next one you're going to write down? It's responsible, so it's in the front. That's nice. It gives you an idea of where it is. It's in the front. It's responsible for motor function. In it, we find the primary motor area located on a single gyrus. Guess where it is? Right next to that other one. It's called the precentral gyrus. Controls basic skeletal movement. Once again, a volunteer went under the knife, and we could touch parts of this, and it would cause part of their body to move. Okay? So that's all controlled right there. Basic skeletal movements. The premotor area is when we have pre-planned skilled motor movements, planning order and frequency of action potentials. Remember how the action potentials have to fire and they have to reach a threshold and all that kind of stuff. So right now your brain knows that you're writing, and so it's getting ready for, oh yeah, pencil's going to be like this, it's going to be going across, I'm going to use these muscles in my arm, I'm going to be using these muscles in my eyes, and that kind of thing, okay? There's a place called Broca's area. It coordinates detailed sequencing for fine motor muscles associated with speech. Now we believe that speech, for what you're going to say, originates in Wernicke's area. Okay, that's that part we talked about that was in the temporal. Was that the temporal lobe? The Wernicke's area. And so now we're going to take that information. The Broca's area is going to be used to coordinate all that. So I want y'all to say Irish wrist watch. Irish wrist watch. So our Broca's area, our Wernicke's is going, I know how to say Irish wrist watch, but your Broca's area is going, Irish wrist watch. Yeah, so it's those tongue twisters kind of a thing right here. All right, and then we also have our olfaction area is found here, deep inside the frontal lobe. What's olfaction?
Nose.
The sense of smell. The sense of smell is found there, okay? And also that would include the idea of remembering a smell. And sometimes, does this happen to you? You'll smell something, it'll specifically take you back to something. And you'll say, oh, I remember as a kid, and I used to smell cinnamon rolls, reminds me of Christmas, something like this. I have one of my grandmothers, she had this little tin with a little, like, perfume. Not liquid, it was like balm kind of a thing. And when I smell it, I just go right back to my grandma's house. Pretty cool stuff right there. That's all found in our frontal lobe. So here's the frontal. So the whole thing here is called the frontal lobe. Sorry, yeah.
Our thing's not working.
And then we're going to see here, when we talk about that speech, okay, that speech, remember, originating in Wernicke's, and then then being translated through the Broca's. All right, there's this thing called aphasia, where you have difficulty speaking, possibly reading and understanding, or writing. Okay, so a language disability usually caused by a brain injury. I know it happens sometimes in little children as they're growing. Maybe things aren't developing as quickly in parts of their brain, and so they have this problem. So here we have our Wernicke's over here. Is that it? Yeah, Wernicke's on the right, orange, over to the Broca's. So it has to communicate to the Broca's area right there to do this. So we can have two types. Broca's aphasia will cause sentence structure to compose of verbs and nouns only. Catch, train, home, now. Speech patterns, it requires effort. It's slow, drawn out, but it means that you can understand what the person is saying. And the person would possibly, they know what they're saying. So let's say somebody's had a stroke, and that part of their brain was affected, and now they're speaking to you slowly with just verbs and nouns, and it frustrates them, and sometimes it might even frustrate you, but you understand what they're saying, okay? Understanding here, sometimes sequency, dog bitten by man, they might get very confused. They might think you said dog bitten by man versus a dog bit a man, kind of a thing. That's the Broca's aphasia. We also have Wernicke's aphasia, where sentences are mostly function words. For instance, I called mother on the TV who did not understand the door. So it's fluent speech, but it doesn't usually mean anything. It's poorly sequenced. Victims don't really know they're doing that. They think they're saying exactly what they want to say, and therefore they would have difficulty with the understanding. Let's watch. This is a patient who has, I believe, Wernicke's aphasia. It doesn't tell us there. Just says
Hi, Byron. How are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment and talked with the people for them over there. They're diving for them at the moment. But they'll save in the moment. He'll have water for him. With luck, for him.
So we're on a cruise and we're about to
We
get
will
to June.
store it right here and they'll save their hands right there for them.
And what were we just doing with the iPad?
Right at the moment they don't show a darn thing.
The iPad that we were doing. Here?
I'd like my change for me and change hands for me. With Happy. I would talk with Donna sometimes. We're all with them. Other people are working with them. I'm very happy with them.
Good.
This girl was fairly good. And happy, and I play golf. And hit other trees.
All right, so he's so cute, right? A stroke victim, he had a stroke, and so he's dealing with his aphasia at this point. So notice some of his things, he wasn't making any sense on some of his things, but he didn't realize he wasn't making sense. Okay, so the person working with him, by the way, was a speech therapist. Speech therapy is an amazing science. I think it's fantastic. It's more than just teaching someone how to make words. It's teaching someone how to use their brain again, how to recognize things. Okay, the prefrontal lobe, that's in the very front of the frontal lobe. This is where we think your personality is found. Reason, so a lot of our reasoning will take place here. Deduction, deductive reasoning, right? You figure something out. Motivation and our aggression, which brings us to something called a prefrontal lobotomy. Okay, it mentioned it in the video we saw earlier how prefrontal lobotomies were fairly common. They're not really common anymore. In fact, I don't know that they do them at all. Okay, the idea here was that this aggression part right here, if you had somebody who had aggressive tendencies, then maybe you can do something to their brain to disconnect that aggression from the rest of it. And so what they did were these things called lobotomies. And a lobotomy means to take the lobe and separate it from the brain. Okay, so they're going to take that front part of the lobe and they're going to try to separate it from, there goes the pituitary, separate it from the rest of the frontal lobe. Okay, the problem is we don't really know where the prefrontal begins, ends, and that kind of thing, right? So I will tell you that there were plenty of successes that took place doing a prefrontal lobotomy. But it's going to creep you out a little bit. The way that it was done is that the skull of your brain is very thin behind your eyes. And so they had these flat sort of metal things that look a little bit like a skewer you use on the barbecue. You know, the flat ones, the metal ones. And they would put them in through your eye socket. Now you're asleep at this point. And it comes up through both sides like this, through the bone, and then they swish back and forth like this. And it just separates the prefrontal lobe from the frontal lobe, all right? So some people who had been dealing with some terrible aggressive tendencies, it would work for them.
It would like not make them as like.
Yeah, but it tended to also change their personality, where they're a little bit kind of like, yeah, you know, this kind of thing. So there's a story, I believe you'll read it in your book, where one of the children of the Kennedy family, the oldest daughter of the Kennedys, so we're talking about Joseph P. Kennedy, the parents of John F. Kennedy, the President of the United States, yes? And their oldest daughter, she had some trouble. She was probably a little mentally behind. You know, she wasn't really bad. She was able to read and write, and she had conversations and things like this, but she would get very frustrated very easily if someone was not catching on to what she was saying, and she would kind of act out, sometimes aggressively, throwing things, trying to hit people, this kind of thing. So the father, Joe Kennedy, wanted all of his children to at least look like they were perfect in public. They had a very high-profile life. And so he, without his wife's knowledge, took her in, her name was Rosemary, took her in for a frontal lobotomy. And when they did it, it didn't go well. In fact, it turned her into a vegetable. In other words, it gave her such bad brain damage that she was unable to do anything. She basically lived in a vegetative state for the rest of her life, which was a long time. She lived to be fairly old. But the wife, Rosemary Kennedy, I don't think ever forgave her husband from that because she was functioning okay. Today, instead of doing these kind of lobotomies, we are working with medications that would try to deal with things, you know, schizophrenia and bipolar disorder, these kind of things. We're looking through medications that can cross the blood-brain barrier and help with that. So fortunately, we don't have it sort of like that. So there's that prefrontal area right there.
What exactly is bipolar?
You know, honestly, I'm not an expert on it. It's a psychological event where you're either really, really happy or you're really, really down. And it's not really in between. Yeah, so you see people who are ready to party and people who you don't even want to be around because they're so depressed. That's what bipolar is. That's my understanding of it, at least.
Yeah.
All right, so remember we talked about how neurons do not go through mitosis, but we still have things we have brain cancers out there. So these are the kind of brain cancers that you can, that are most common. So the gliomas usually happen in astrocytes or oligodendrocytes. So you can see here astrocytoma, oligodendroglioma. So gliomas, those are in some of our microglia, right? So our microglia cells. The meningioma, that's of the meninges. We'll get to those. Those are the linings of the brain. And then we have, here's the astrocytoma again. That is a childhood tumor that develops in children. And notice where it is. It's in the cerebellum. So what might be a symptom of somebody with a tumor in their cerebellum?
Like acting.
Remember what cerebellum does?
Is it like the functioning muscles?
It would be a balance type of a thing. Yeah, their coordination might be off. Here we have this is called the pontine. We're going to see the pontine in a minute. That's inside the brain stem, right? So there would be some trouble right there. Here's one called the schwannoma, Schwann cells, right? A tumor of the Schwann cells. Ependymoma, whatever. That's the in some of those, I call them ependymal cells. They would have a tumor there. Okay. Now in the middle, this is a blank you're filling in that says what's inside the center of the brain. You're going to write the corpus callosum. Corpus callosum. Right in the middle, this kind of large thing right here of all white matter. It's the main commissure, the main thing that happens between the right and left hemisphere, allowing for communication between the hemispheres. All right, so I have the two sides of the brain, my left side and my right side of the brain. So here's this guy. I only have one eyeball. He's my pirate, right? Only one eye. So we take it apart. We can see there's our brainstem. So we have our right side and our left side of our brain. Okay, so it's interesting how with that is that the right side, which controls the left side of the body, tends to be the side that's associated with things like artistic abilities, music, sort of those abstract kind of things there. Some of the greatest artists in the world are left-handed people. Some of the great musicians are left-handed people. Not all of them. In fact, we're the only animal out there that shows a preponderance toward one side or the other, and it's overwhelmingly right-handed. we're like in the 90% right-handed people. And so left brain controls the right side, and then that would be for things that are more like reason, math, those kind of things, not the artistic things. And so I don't know about me, my math is not good. So, but what's kind of interesting in that corpus callosum, it shows it here, it's kind of pink still right there, is there's this crossover that takes place. All right, where the right side of the brain knows what the left side of the brain is thinking. Yeah. So it turns out though that women have more connections between the left side and the right side of the brain. Okay, so the right side, the artistic and emotional side versus the logical side over here, they have a lot more ties there. Apparently when a male is in the womb, there's a time when he's bathed with testosterone and it breaks a lot of those ties between the two sides of the brain. And so how does that manifest itself? Well, it manifests itself with women. For instance, if you've ever had an argument with somebody and you're really mad about something and then you start to cry about it, that's because our emotions are tied in, right? And the men are going, ugh. So also think, the way I like to think about it is this. You're picking teams, right, for your baseball team. And so you're looking out there at everybody and you see that little guy that nobody picks because he's short and you're like, oh, I want to put him on my team. Whereas your dad's picking a team going, I don't want that kid on my team, he's no good, and has no emotions at all. And so we tend to be more affected by emotions when we're making decisions, where men tend to make decisions without much emotion involved because their brains aren't as connected. Okay, do you get it?
Yeah.
Is
Yeah.
it true? I heard the other day, I've heard this before, that like women or girls are typically born stronger than men.
Physically stronger?
Yeah.
Oh, I don't know. I know our upper body strength doesn't match theirs at all. Not even
Or
close.
girls are like physically born stronger or just healthier than males typically. Stronger and healthier.
Well, what I do know is that, think about it. This helps women to be more motherly, where they can deal with their children with some emotion involved, whereas the dad can be the disciplinarian without a whole lot of emotion involved. Does that mean that men don't have any emotion? Of course not. Of course not. They definitely have crossover. It's just not as much. So we tend to let emotion make decisions. Men tend to let logic and reason make decisions. Okay, but that doesn't mean that we don't. But there is a difference between male and female. Is what I want you to know. Okay. All right.
So can you look at a brain and like if it's male or female?
No, not by looking at it. No. It's mostly made of white matter. The left brain tends to be for speech, math, and reason. The right brain tends to be art, music, depth perception. That's interesting. Have you ever heard that women are better at multitasking?
Yeah.
This is why. This is why. The men, they just want to get this thing done. I'm just going to get this done right here. Don't bother me. I'm going to do this thing. We're like, I can do it all. I used to be a better multitasker than I am now. But okay, that's our corpus callosum. Now, those lumps of gray matter that we find embedded in the white matter. So we're talking about the brain stem. We're talking about other parts of white matter. Okay. So this is what you're going to write down. Other important structures in the brain, you're going to write basal nuclei. And if you want to say ganglia, basal nuclei or basal ganglia, it's called both things right there. This is lumps of gray matter, so it's going to be
all the like points.
right here, lentiform nucleus, caudate nucleus, subthalamic nucleus, and substantia nigra. All right, so these are gray matter embedded in white matter, okay? So this will help with certain things like planning, initiation, maintaining and concluding motor actions, muscle tone, posture, all these things right there. And we've noticed a few things about these inner structures inside the white matter. And I'm going to show you pictures of it here in just a second. And remember that my problem is, y'all, I don't think I'm going to get through this lecture in time if I stay too long on each one of these slides, okay? Which means I'll either have to keep you late. You don't want to do that? Okay, then we're going to move on. So here we're looking at them. You can see right here in the middle of, so here's the brain cut in half. You can see on the left side here. And you can see these colored things over here. So notice we have the caudate nucleus, kind of that purple color there. This blue right here is the thalamus itself, okay? So the thalamus is, we know it's in the middle right there, right? We have the lentiform includes how do you say that word? The putamen, and the globulus pallidus. These two together are the lentiform. and our subthalamic, below the thalamus nucleus, and our substantia nigra. Nigra means dark, so gray matter, dark matter, that dark stuff. And so that's the stuff, so it's doing some thinking there. These are those basal ganglia. And I want you to particularly look at substantia nigra here's our lentiform, and here's our substantia nigra right here. There seems to be a connection between that and Parkinson's disease and the shrinkage of that little nucleus inside there. So if we look at it here, this part here tends to shrink up. We've noticed that in Parkinson's patients. Oh gosh, I don't know if we have time for this.
How do you notice something like that? Like
Brain scans.
it's such like a small area.
Yeah, they use a brain scan.
Parkinson's is the second most common neurodegenerative disease after Alzheimer's. The most visible features of this disease are a slowness of movement, muscle rigidity, and tremors at rest. There are also many, often debilitating, non-motor symptoms, some of which may present before the motor problems. Parkinson's affects many areas of the nervous system and different types of neurons. However, much focus has been placed on neurons in brain regions associated with the overt motor symptoms, notably a region of the midbrain called the substantia nigra pars compacta. This region forms part of a major pathway in the brain that's critical for facilitating movement. In Parkinson's, dopaminergic neurons in the substantia nigra gradually die, leading to the malfunction of this pathway and the characteristic motor problems. Drugs that replace or mimic dopamine are often used to treat these motor deficits, but they grow less effective over time. Deep brain stimulation may also be used to treat symptoms. However, no current treatment slows the neurodegeneration. A distinctive pathology in most cases of Parkinson's is clumps of misfolded proteins within neurons. Lewy bodies are the most common type. A characteristic component of these is a misfolded protein called alpha-synuclein. These molecules can form small repeated units called oligomers, or longer fibrils. Mounting evidence indicates that these are toxic to neurons and play a key role in driving Parkinson's. Unwanted proteins are normally cleared by the cell's different types of protein-degrading machinery. Some evidence points to these systems being overwhelmed by misfolded alpha-synuclein, and this might be a key factor in what kills the neurons. Parkinson's has also been linked to problems with mitochondria. Mitochondria provide cells with the energy to perform vital functions. They're highly dynamic and can fuse together or break up into smaller versions in response to a cell's energy demands. They can also be transported to areas of a cell that need them the most. However, in Parkinson's, these processes can be impaired, and mitochondria are unable to sustain proper neuronal function. As they become old or damaged, mitochondria are removed and replaced. Again, this recycling is thought to be disrupted in Parkinson's, leading to the accumulation of damaged or worn-out mitochondria. Another idea is that the cells called glia surrounding neurons may play a role in Parkinson's. As dopamine neurons are lost, one particular type of glial cell, called microglia, is thought to take up the resulting cellular debris, triggering an immune response. Once activated, they release inflammatory cytokines, which activate neighboring microglia and another type of glial cell called astrocytes. Chemicals released by activated microglia and astrocytes have been shown to injure neurons. It remains unclear which mechanisms drive the disease process in Parkinson's. What is clear is that with time, more areas of the nervous system develop pathology. And one emerging idea is that this happens through the transmission of misfolded alpha-synuclein. While the multifactorial nature of Parkinson's disease progression might make it hard to fully understand, it does provide a number of potential therapeutic targets for slowing or even halting the neurodegeneration. For example, enhancing the clearance of abnormal proteins and blocking their transmission, improving the function of mitochondria, and targeting the neuroinflammation are all currently active areas of research.
Okay, so we're gonna move on. So the next box you're gonna It says here protection of the brain, ventricular system of the brain. Cross that out and write down limbic system. These are nuclei and commissures in the cerebrum and the diencephalon. They influence mood, emotion, fear, pain, and pleasure. Our survival instinct is found here. Remember, these are in the clumps of gray matter. The amygdaloid nucleus remembers emotions. This is the part of your brain that holds grudges. So if you remember you're mad at somebody, I don't hold grudges for some reason. I don't. My amygdaloid's not very pronounced, I guess. I'm mad at my husband, and I'm like, oh, I'm gonna be mean to him, and then I forget, and so I'm nice to him. Bummer, yeah.
Oh my gosh,
so.
I do that all the time when I get mad at my siblings. I'm like, I'm not doing anything nice to you, and then like 5 minutes
Then you
later,
forget.
like, I love you.
Yeah, right. So that's how I am. It's good to not hold
I
grudges.
get mad when they don't talk to me, so I'm like, I'm gonna talk to you.
All right, remember, we're going to move on. Sorry, y'all. Here's our amygdaloid right here, amygdala, right in there. All right, and right there, the red part. Okay, part of the limbic. Did you get it? Well, I have this whole thing recorded for you.
Okay.
Send the whole thing to you. All right, the ventricles are inside the brain. I think that you're going to turn the page now. And you're going to, at the very top, you have 2 lines, write ventricles of the brain. All right, they're cavities. These are cavities. These are holes in your brain. Okay, holes in your brain. There are 4 of them. And their job is to produce cerebrospinal fluid, CSF. It cushions your brain. It provides nutrients. And it's lined with ependymal cells or ependymal cells. I call them ependymal. Ependymal. They have those little ciliated ones that cause this cerebrospinal fluid to be moved. So inside the middle of your brain are the ventricles. They produce the CSF and they squeeze it out. I guess they squeeze it this way, around the front, over the back, and then it ends up draining all the way down into the spinal cord here. It's constantly being made and constantly being pushed back, okay? It's keeping that space. Have you ever heard of a spinal tap? Sometimes if there's something wrong with you, they want to take some of your cerebrospinal fluid out to test it, to see if there's anything wrong with it, mostly to see if it's got any kind of bacterial infection in it or something. And what they'll do is they'll take it out, it leaves a little void in your that space. And so you have to lay down until there's time enough for that cerebrospinal fluid to refill that space. They don't let you get up out of bed or anything. It's a hospital procedure. There's a story, I think, in your book about somebody who got up too quickly and boom, down they went because it just caused that brain to almost be like stuck together, if you will. There's not that cushion that's being created right there. When you do your, dissect your brain, you're going to see those holes very easily, in fact. Okay, so the meninges are the 3 layers of tough membranes around, that's the next box. It says sketch and label the ventricles. Right there, you're going to write down meninges in that box. I'm not going to make you sketch and label the ventricles of the brain.
It says what are the meninges of the brain?
That's good, all right? But in that box above it, write meninges, okay? Because I want you to write there's three types of meningeal membranes, these membranes. The first one's called the dura mater. The dura mater. That means tough mother. Tough mama is what that means. It's firmly attached to the bone, reaches down through the dural sinuses. Okay, so remember, this is the protection of the brain. It's like a coating over the brain. The arachnoid matter. Arachnoid meaning having to do with spiders. It's because it has web-like tissue that allows the cerebrospinal fluid to flow around it. And the pia mater. Pia mater means to hug. And it's tightly bound around the brain surface. It hugs the brain surface. The brain, when you get it to dissect it, it will have the pia mater on it. You won't be able to tell, really, because it's hard to separate it from there. Okay, and the inflammation of the meninges is called meningitis. Have you heard of it? Meningitis. That can happen a couple of ways. Viral meningitis, and bacterial meningitis. I always wondered why bacterial meningitis was worse than viral, because typically with the bacterial infection, you can take an antibiotic, right? With the virus, you just got to wait. Well, it turns out with bacterial meningitis, we create pus, and that pus will fill that area, possibly pushing against the brain, possibly causing brain damage. And so bacterial meningitis is worse than viral. Viral doesn't have the pus produced, therefore with viral, you just have to wait and have time to become better. Okay, this is one of those deals people say if you try to turn your head and it hurts so bad, sometimes that can be a meningitis situation going on there. All right, here's a picture of it. Here is our dura mater, very, very tough, thick layer right there. Then the arachnoid mater, you can see, is looks like a spider's web right there. And then right hugging the brain is the pia mater, right there. Here's another picture from this side. All right.
This is just the layers.
Yeah, those are the layers. Yeah, so here's what we need to do, y'all. Our cerebral spinal fluid is constantly produced, distributed by pressure and ependymal cells, squeezes through the arachnoid matter, flows to the spinal cord, eventually dumps into the duracinus, a huge vein that mixes into the bloodstream. The subarachnoid space must be continually filled with CSF, and that spinal tap safety is about laying down until the doctor says you can get up. If you have bleeding in the subdural place, we call that a subdural hematoma. Now we're finally at 8B. Look what time it is. We're just now getting to 8B. Okay, here we go. The spinal cord. Communication link between the brain and the peripheral nervous system, the brain's
Only one page,
ponytail.
though.
Yeah, there's only one page, and this stuff at the beginning is just introduction. There's no place to write this down. There are 31 pair of spinal nerves which exit through the vertebrae. 2 bulges, one called the cervical enlargement, which is where it comes out for the upper limbs. 2, the lumbar enlargement, where the nerves come out at the lower limbs. The bottom of the cord tapers to the conus medullaris, also known as the horse's tail or the cauda equina. It's the horse tail. We're going to see a video here in a minute. All right, this is what you're going to write in those boxes. Ready? 7 cervical vertebrae, C1 through C7. These top red ones here on the left side. Your next box will say 12 thoracic vertebrae, which is T1 through T12. These purple ones on the left side. 5 lumbar, L1 through L5, the green ones. Below that, the blue part is called the sacrum. And that little tiny bit on there is called the coccyx or the tailbone.
My grandma was in a really bad car accident almost a year ago, and she like broke or did something to all of this stuff. So it's so funny because I'm like so familiar.
She broke her back.
Yeah, she broke.
Not her neck, I hope.
No.
Okay, she's
she
not
did a
paralyzed.
ton of stuff to like her back. And,
Okay.
Oh yeah, no, she's totally fine now.
So keep in mind, I'm going to show you a picture of the spinal cord runs through those vertebrae, right? So if you have an injury in these red ones up here, C1, if you have an injury right here at the top at C1, you're dead. C2, you can die, but you can survive with that. Below that, you're talking paraplegia or quadriplegia, paraplegic at C7 or so, C6 maybe. And further down here, you tend to not have paralysis, but you do have certainly trouble with a broken back, if you will. This is what the tissue looks like. We have some gray matter and some white matter, right, in our actual spinal cord, right here. And they all have these names. You'll see that in your book. Dorsal horn, the lateral horn, this kind of thing. The reflex arc right here goes from the spinal cord to the effector muscle right here, like say the touching of the skin. In other words, you know how we're talking about reflexes? It doesn't have to actually go through the brain. It can actually have its communication right there through the spinal cord. It's quick, it's responsive. All right, if we're looking at it, here's our vertebrae. The disc is a bit of cartilage. This yellow thing coming down, the big fat one, you can see at the top and the bottom, is the spinal cord, and then the little nerves that are coming off the sides. If the disc starts to bulge, it will bulge and pinch that spinal nerve right there, causing that pain that people have if they have a pinched disc, right? And sometimes it squeezes that disc out and you'll have bone rubbing together, which will also cause pain. Some people will have artificial stuff put in there to replace that disc. Our neural pathways, these are your next 2 blocks, right? We have ascending and descending pathways in the spinal cord, and it refers to, you have the sentence to finish right here. It refers to motor and sensory pathways where information travels from the brain to the PNS and back. So you're going to put that in that blank as well as you can. All right, so motor and sensory pathways, where information travels from the brain to the PNS and back again. This is all about getting information from the sensory and to the sensory, right? I feel something, I move. And you can see it's traveling down through the parts right here. So we have 2 pathways. We have the ascending and the descending. So let's go through each one of those really quickly in those two boxes right there. The descending. So this says figure 812 right here. It's called the motor pathway, the descending motor pathway. A decision to move a muscle begins at the brain, descends to the muscle moves. That's all that is. And I think all I want you to do is write those words in there, okay? I think they wanted you to draw it, but you'll see it in your book, figure 812. So if the motor pathway is descending, that makes the sensory pathway ascending. That makes sense, doesn't it? It's going to go from my sensory organ to my brain. Notice this travels down, crosses over, down to the right side of the brain, or the right side of the brain to the left side of the body. Our ascending sensory pathway. Sensory information is gathered by the senses, sent up to the brain for interpretation. That's the ascending sensory pathway. So if you call it descending motor pathway and ascending sensory pathway, you're going to remember that. No, it's not that hard, though. It makes sense. The brain tells the muscle to move, so it's going from the brain to the muscle. That's descending. The sense tells the brain to do something, or gives information for the brain to interpret, and therefore it's going to ascend. All right, and we have a little video. I want you to see our spinal cord.
What we have here are two human brains that we got from cadavers a number of years ago and have had in storage in the lab here. This one, of course, has a spinal cord. I would like to show you the spinal cord now. It's a continuation. I sometimes call it the ponytail on the brain. You can see it continues down. This is the anterior side. It's a little bit flattened. That is, it's a little wider left to right, a little flattened front to back. And it has the same coverings, same dura, same arachnoid, same pia that the brain has. We can only see the dura mater right here. The other layers are there. You can see projecting from each side, right and left, are the spinal nerves that come out. Their roots are right at the cord, and then they will come out, course through the body, go where they're going. Here we can see them. This is called the anterior root and the posterior root. Interestingly, all the motor output, that is the neurons that send messages to muscles, will be located anteriorly. On the other side, you see the roots coming back in the back here? Those all contain sensory information. They're taking action potentials that way, the ones in front. So information goes out the front and in the back.
Out the front, in the back.
Those are our roots. That's a sensory root. That's a motor root. They'll go together through the body and form a spinal nerve. Here's something really interesting. Did you know that the spinal cord ends sooner than you think?
The horse's tail.
And it ends in coarse groups of neuron axons. And this together is called the cauda equina, which means the horsetail. And it does look something like a horsetail. And it's finally anchored in the sacrum by the connective layers. That's our cauda equina of the spinal cord.
All right, that's it. Okay, gang, I'm done. We made it. Look, I just kept you a minute late. Yes, anybody who wants the video, let me know. I will send it.